Uh, Good afternoon. My name is Katie Politi, and I'm an associate professor of pathology and of medicine here at the Yale School of Medicine, and also I'm co-leader of the Cancer Signaling Networks research program at the Yale Cancer Center. And it is my pleasure to introduce Dr. Michael Shen today. Dr. Shen is a professor of medicine, genetics and development, urology, and systems biology at Columbia University Medical Center. He received his undergraduate degree from Harvard University and his PhD from Cambridge University. He then pursued his postdoctoral training with Dr. Phil Leader at Harvard Medical School before becoming an independent investigator at Rutgers in 1994 and moved to Columbia University Medical Center in 2007. He is currently the co-leader of the Tumor Biology and Microenvironment program at the Herbert Irving Comprehensive Cancer Center, as well as the Director of Graduate Studies in the Columbia Department of Genetics and Development. Over the past 26 years, Dr. Shen has investigated fundamental mechanisms of mammalian development and cancer using in vivo analyses of genetically engineered mouse models. Recently, his lab has generated novel culture conditions for mouse and human prostate and bladder organoids, which have led to the creation of a biobank of patient-derived bladder tumor organoids. Current work in the lab focuses on understanding molecular mechanisms of cell type differentiation in the normal, as well as the transformed prostate epithelium, the epigenetic regulation of lineage plasticity in both bladder and prostate cancer, and the role of the tumor microenvironment in modulating treatment response. Dr. Shen, it really is a pleasure to have you here today and have you visit, albeit virtually, from Columbia University. So thank you very much, and we look forward to your presentation.
Well, thank you, Katie. It's a real pleasure to have this opportunity to speak to this audience at Yale, and I wish this were in person, but we will make do as best as we can. So I'll go ahead and share my screen. And hopefully you can see my presentation. Is that true? Can everyone see my?
Yes, we can see it. Thank you. Yes.
Okay. Excellent. So today I'd like to take the opportunity to discuss published work, but also a lot of work that can be construed as work in progress. Much of it focusing on the issue of lineage plasticity. And we've been studying this through in vivo analyses in mouse models, but also using organoid models. And we've been studying this in both the prostate and the bladder. So to start with, what is plasticity? So if we consider that plasticity in the most general definition is the ability of a cell to change from one identity to another, we can think of this in the, perhaps cliched Waddington model as, sort of balls rolling down a hill. We start with a stem cell and we have various differentiated cell types, and the ability of cells to basically change their identity is considered to be plasticity. And so there are different forms. They're sort of reprogramming back to a more progenitor state. There can be transdifferentiation, changing from one identity to another, etc. So this is a process that has been studied extensively in both development and cancer. And it's important to think about, when we talk about plasticity in cancer, to consider that in order to study plasticity, it's also essential to understand the normal pathways of differentiation so that one can distinguish what happens in the normal context from what might happen in a tumor context. So over many years, we've been studying these issues in the prostate and more recently in the bladder. And to start with, I'd just like to consider sort of a basic review of the prostate. In the mouse, the prostate has a distinct anatomy. It sort of has a lobular structure. There are 4 different lobes in the mouse. There's the anterior prostate, the dorsal prostate, the lateral prostate, as well as the ventral prostate. However, in the human, there is something a little bit different. The human prostate does not have a distinct lobular structure. Instead, it can be distinguished at the pathological level as having sort of architecture of different regions. So there are zones that have been defined pathologically, the peripheral, central, and transition zones. So the distinct anatomy of the mouse and human prostate has been of note for many years and has been used sort of as an argument as underscoring the inability, perhaps, of the mouse to truly model human prostate cancer. Now, we've known from studies over many years that there are many conserved signaling pathways and the like, but the relationship between the mouse and human prostate still remains somewhat mysterious, both in terms of normal development as well as the cell types contained within the prostate. Now, the prostate in the mouse is formed at late stages of fetal development, and organogenesis primarily takes place at neonatal stages. The prostate forms through a process of ductal budding from the urogenital sinus, and initial prostate buds are marked by expression of the homeobox gene NKX 3.1, here visualized by beta-galactosidase of a LAC-Z knock-in that we made a number of years ago. And you can see even early on at the time of birth, there are buds that correspond to distinct lobes, and at least initially, NKX 3.1 is expressed by all of the epithelial cells in the prostate. Prostate formation, of course, requires androgen signaling, but the requirements for androgen receptor are actually fairly complex, and they involve epithelial mesenchymal interactions. So androgen receptor is actually required in the urogenital mesenchyme for prostate formation. So if you delete androgen receptor in the urogenital mesenchyme, here in a TFM mutant, testicular feminized mutant, and you perform a tissue recombination assay, as was first shown through the studies of Jerry Cunha about 4 decades ago now, the prostate will not form. However, if you delete androgen receptor in the epithelium, now you will form a prostate. However, the prostate is not entirely normal. For example, it lacks secretory protein production. So what are the cell types that are found in the normal adult prostate? Well, in both the mouse and the human, there's an array of different cell types in both the epithelium and in the stroma that are just now really being characterized in some detail. But historically, we've considered the epithelium as containing 3 basic cell types. There are the luminal cells, which are the tall columnar secretory cells that produce the prostate secretions. There's an underlying layer of basal cells. These cells are androgen receptor low or negative and express basal cytokeratins, unlike the luminal cells, which are AR high and express luminal cytokeratins. And then there is a rare third type known as neuroendocrine cells. These have been very understudied, and I'll touch upon these later in my talk. So this is sort of the way we've thought about the prostate epithelium for many years now. And classically, basal cells have been considered to be more interesting. They appear to have more stem-like properties, whereas luminal cells have been considered to be somewhat boring. But I think, for many years, we've thought that luminal cells are actually the interesting cells. And now with the advent of single cell analyses, we see that there's considerable complexity in the luminal population. So what do basal cells do? Well, we believe that there is sort of a conserved ancestral role for basal cells, and that's in wound repair. So here, for example, in the prostate, if we damage the luminal cells by deletion of E-cadherin, inducible deletion of E-cadherin. The epithelial cells will slough off and undergo anoicus, and the basal cells will actually differentiate into luminal cells to replace the lost luminal cells. So this is shown in cartoon form here. So there's sort of a basal to luminal differentiation that takes place. So we think that this is a conserved function of basal cells in many tissues. And more recently, work from Cedric Blancpain's lab has demonstrated that is in fact the case. What about luminal cells? Well, luminal cells, as I mentioned, have been considered to be somewhat boring, but in fact, in ex vivo assays at least, one can see that there are luminal progenitors that are bipotent. So if we perform lineage marking of luminal cells and generate organoids that are wholly composed, wholly derived from luminal cells, then we can see that there are basal cells that can be formed in these organoids, and that these basal cells are marked, indicating their luminal origin. So, of course, this is an ex vivo assay, and ex vivo cells often display more plasticity than is found during normal development. And certainly in normal contexts, luminal cells are usually unipotent. But we think that normal developmental processes, among other things, can be constrained by the microenvironment. And ex vivo assays such as organoid formation can reveal developmental potential that can be displayed in specific contexts. So, for example, we think that the ability of luminal cells to display bipotency, to be able to generate basal cells, is actually an ability that occurs early in organogenesis. So this is sort of a lineage hierarchy as is currently understood, in which there are bipotent basal progenitors generating both basal and luminal cells during organogenesis. But luminal progenitors were generally thought to be unipotent, but in recent studies, we found that in fact there is a bipotent luminal progenitor. So if we lineage mark luminal cells at early postnatal stages, using an inducible CK8 driver, we can then mark luminal cells, and then analyzed later, luminal cells are marked, but also basal cells. And it's a reasonable fraction of basal cells that are marked. And this bipotent progenitor is fairly short-lived. It's fairly transient, but it can still be detected at a week after birth. But again, the ability of this bipotent progenitor quickly disappears thereafter. So interestingly, both the bipotent basal progenitor and this bipotent luminal progenitor do not require androgen receptor function. So if we delete androgen receptor in basal cells, we find that there's no effect on the formation of luminal cells. And similarly, if we delete AR in luminal cells, we don't see any effect on the generation of basal cells. So in cartoon form then, what we think is going on is that there is a urogenital epithelial progenitor that gives rise to both basal and luminal progenitors. And initially, the basal progenitor can be bipotent as well as the luminal progenitor, but that this bipotency is fairly transient. And then in adulthood, both the luminal and basal progenitors are generally unipotent. However, this period of bipotency is actually occurring in the first 4 weeks after birth. And interestingly, this is a time when androgen levels are very low at these prepubertal stages. Now, there's one other interesting aspect of luminal cells, which is that in a series of studies, we've shown that they are favored as cells of origin for prostate cancer. So we've shown this in a number of different transgenic mouse models, as well as a hormonal carcinogenesis model. If we mark basal cells in these models, for example, in the high-MIC model or a TRAMP model, which are well-characterized transgenic models of prostate cancer, the basal cells are marked, but they don't really contribute to tumors. However, if we mark luminal cells, now they readily contribute to tumor formation. So what we think is going on then is that if we mark luminal cells, they'll contribute to tumors. And this argues that luminal cells are a cell of origin for prostate cancer. If we mark basal cells, they don't contribute to tumors. However, if you explant these cells, they will, in this sort of ex vivo context, for example, in a graft, undergo a basal to luminal differentiation, and now they can form tumors. So we consider basal cells to be a cell of mutation, whereas luminal cells are the true cell of origin. And again, we've analyzed this in multiple mouse models of prostate cancer. So because we've observed the same result every time, we think that luminal cells are generally favored as a cell of origin. So we now have this view that luminal cells are in fact quite interesting. So recently we decided to explore the heterogeneity of the prostate epithelium using single cell approaches. For this, we had to really learn how to dissect a mouse prostate properly. You might think that's a bit of an exaggeration, considering we've been studying the prostate for over 20 years. But in fact, it's really not a trivial matter to dissect the individual mouse lobes all the way down to their junction with the urethra. So this is sort of a view of sort of how we can dissect away the mouse lobes. This is actually in a green mouse, so you can see the lobes that are dissected. And then we subjected these to single cell RNA sequencing, both the whole prostate as well as the individual lobes. To analyze the results, we collaborated with Raul Rabadán's lab. Raul has developed amazing algorithms that are based upon rather sophisticated mathematical approaches for analyzing single cell data. So in particular, his laboratory has developed approaches based upon random matrix theory, which demonstrate that these large arrays of data, for example, as one generates using single cell RNA sequencing, are mostly noise. So if you consider these as giant matrices, well, in fact, the distribution of eigenvalues follows a sort of conserved mathematical distribution known as the Marchenko Pasteur distribution. And the deviation from this distribution is actually where the signal is. And typically it's only about 2 to 3% of the data. So this is a hypothetical distribution. This is actually real data. This is one of our prostate data sets. And again, here is the signal. So his laboratory has developed randomly an algorithm to isolate these data. And this proved to be very useful in our analyses because it allowed us to identify a cell population that would have been very difficult to identify otherwise. So when we examine an aggregated data set of whole prostate, we observe 5 different luminal populations. First of all, we only identify one basal population. So basal cells are actually not heterogeneous, but instead there's heterogeneity in the luminal population. So first of all, there's 4 distinct luminal populations that correspond to the, that are located distally in each prostate lobe. So there's a Lum A population distally in the anterior prostate, Lum D in the distal dorsal prostate, Lum L in the lateral, Lum V in the ventral. Then there is a population that we call Lum P for proximal. It is similar in all 4 lobes, and it is found more proximally. Finally, there is a population that we call periurethral. This population has both basal and luminal properties, and it is found in the region most adjacent to the urethra. So these are the distinct epithelial properties that we've identified. We've also identified heterogeneity in the stroma, but I won't speak to that further. So as you can note from this sort of illustration, there is diversity along the proximal distal axis. So to give you an idea of what we mean by proximal distal axis, here's an anterior prostate that's been sort of splayed out and cut in a histological section. When we refer to distal, we're actually referring to this whole region here. That is more than 90% of the volume of the prostate. The proximal region is just this region here. And these regions are quite distinct at the level of marker expression as well as histology. So here, if we go along the proximal distal axis, we have the paraurethral region. It has specific markers. Here's the proximal region. It has a distinct histology and is marked by specific gene, the expression of specific genes, such as this one, PPP1R1B. Then we have in the distal region other markers that are specific for distal luminal cells, the LumA population here, but you will note there are also proximal cells that are scattered about. They're not very common, but you can definitely find them in the distal region. And then in between, there appears to be a boundary where these regions meet. If you perform electron micrography, of sort of the boundary region, you can see that these LumA and LumP cells actually appear to be distinct cell types. So the LumA cells, again, are tall columnar secretory cells. The LumP cells, on the other hand, have a more cuboidal appearance, and they don't seem to be particularly secretory. At the transcriptomic level, you can analyze the sort of relationships of these populations with each other. So Luis Aparicio, postdoc in Raul's lab, who's done these computational analyses, has used an approach based upon optimal transport theory to calculate Wasserstein distances between these populations and then relate these populations to each other. You can see that LumP is sort of at the center, related to the distal luminal populations, and then PRU and basal. So in order to investigate the function of these populations, and so as you might imagine from this sort of relationship, we observe that there's greater progenitor potential in the LumP, PRU, and basal populations versus the distal luminal population. So here's an organoid formation assay. You can see that the distal luminal populations all have a low efficiency of formation of organoids. LumP has a much greater efficiency, and PRU and basal have a greater efficiency yet. We've also isolated these populations by flow cytometry and performed renal grafting assays. So in combination with urogenital mesenchyme, these cells will form renal grafts, albeit with different efficiencies. And then we can analyze the sort of cell types present within these grafts. So using the markers that we've identified that are specific for each population. In brief, the LumA and other distal luminal cells can make more of themselves, but not the LumP and PRU populations, whereas the LumP and PRU populations can make all of the other different populations. So this supports a sort of a progenitor, increased progenitor potential for the LumP and PRU populations. So finally, one can ask, what is the relationship between the mouse and human prostate? After all, anatomically, they're quite different, and histologically as well. So we've analyzed 3 independent benign prostatectomy specimens at the single cell level. And again, we can see that there is a single basal population, but there are different luminal populations that we can relate to the luminal populations that we see in the mouse. So there is an acinar population that seems more closely related to the distal luminal populations, a ductal populations more proximal, as well as a PRU-like population. Again, using analyses of Wasserstein distances, this time in a cross-species way, we can definitely show this relationship. So the acinar cells are actually, interestingly, most closely related to luminal L cells in the mouse, the ductal cells to LumP, and PRU to PRU and LumP, etc. So this analysis highlights LumL as a population of interest in the mouse. It perhaps is most closely related to the bulk of the human prostate epithelium, yet the lateral lobe is understudied in the mouse, and particularly in analyses of cancer models. So now I'd like to turn to cancer a little bit and think about plasticity in advanced prostate cancer. So in the current spectrum of prostate cancer, where we have treatment with potent anti-androgens that are very efficient at suppressing androgen receptor function, now castration-resistant prostate cancer is displaying a range of different entities, a sort of a spectrum that can be distinguished perhaps by different differential lineage plasticity. So there is prostate cancer that remains AR pathway positive. It still expresses androgen receptor despite the presence of anti-androgens. And then at the other extreme, we have neuroendocrine prostate cancer, which is AR negative, and in its most extreme forms can display a small cell phenotype that's very aggressive and lethal. So there's been considerable interest in the mechanisms of neuroendocrine differentiation in CRPC. And so there have been studies of CRPC that are trying to distinguish the different entities. And for example, there is something considered double negative, that is AR negative and neuroendocrine negative, which is defined more by what it is not rather than what it is. But the relationships between these distinct entities is unclear, and it may be simple, it may be more complex, but there is widespread agreement that there must be a role for epigenetic reprogramming in this process. And so a range of studies have provided evidence that there's increase in EZH2 as PRC2 activity in this spectrum of plasticity. And recently, Filippo Giancotti's lab has shown that PRC1 activity is elevated in double negative prostate cancer, et cetera. But this is all involves studies either in cell lines or in human prostate cancer specimens. So it's been difficult to sort of study these things, you know, at a more detailed level and in terms of mechanism as well. So to approach this, we started with a mouse model that we had been analyzing in collaboration with Michael Shen's lab. So the NP-p53 mouse model uses inducible deletion of P10 and P53. These animals develop a castration-resistant prostate cancer that will display features of neuroendocrine differentiation. And we were able to distinguish what we called focal neuroendocrine differentiation, in which the neuroendocrine cells are not proliferative, from overt neuroendocrine differentiation, which often displays a small cell phenotype. These are highly proliferative neuroendocrine cells. However, since we use lineage marking here, we could show that the neuroendocrine cells are derived from a luminal cell. So the initial tumor induction was from luminal cells, and then we have formation of CRPC, neuroendocrine differentiation, and then there is some type of proliferative switch that we don't understand that results in this highly proliferative neuroendocrine prostate cancer. So because we used lineage tracing here, we provided evidence that in fact this was a transdifferentiation of luminal cells to neuroendocrine cells. So when we think about transdifferentiation, there's sort of a fundamental question, both in developmental as well as cancer contexts, which is what is really going on in terms of the pathways that result in transdifferentiation? Well, it's possible that this change in identity occurs through a transition that happens normally during development. Alternatively, it's possible that this change in identity occurs through a transition that is wholly or at least partially novel, that doesn't really occur in normal development. So there could be a hijacking of an alternative pathway or some other pathway or process that doesn't occur in normal contexts. To understand this, of course, it is first of all important to discover how neuroendocrine cells differentiate normally. So it's remarkable that there's very little in the published literature about the origin and, in fact, features of neuroendocrine cells in the normal prostate, likely because they're quite rare. And what is also interesting is that there are different models have been put forth for their origin. So one model says that they actually are of epithelial origin and arise from basal progenitors. Another model is that they arise from neural crest. And so Cedric Bonpin's lab published that they came from basal cells, and a more recent paper from that they were from neural crest. And both of these papers use lineage tracing. In fact, we believe that both of these papers are incorrect, and most likely they arise from an early urogenital epithelial progenitor. So neuroendocrine cells are very rare, but what makes them particularly rare in the mouse prostate is they're highly asymmetrically distributed. So they're mostly found in the proximal region, which I showed you earlier. So nearly all of the neuroendocrine cells are proximal. They're very rare distally. And remarkably, neuroendocrine cells, despite their rarity, are heterogeneous. So about 80% of neuroendocrine cells have a luminal-like phenotype. They actually express androgen receptor remarkably, and they express luminal cytokeratins. And then another 20% of neuroendocrine cells are basal-like. They express basal cytokeratins and p63. They can be detected very early in organogenesis at most, perhaps all neuroendocrine cells are actually formed at prepubertal stages. And since neuroendocrine cells, I didn't mention this on the previous slide, are never divide. They appear to be post-mitotic. We believe that they're made and are not subsequently generated. By lineage tracing, we believe that they have an epithelial origin. So using an NKX-CRE driver, we can mark most of the prostate epithelial cells, and in fact, the vast majority of neuroendocrine cells are marked by NKX-CRE. So we believe they have a prostate epithelial origin. So there's more than 95% of the neuroendocrine cells are marked in this experiment. And finally, neuroendocrine cells, likely arise, as has been shown in the lung, through a proneural pathway in which sort of the master regulator at the top of the sort of transcription factor hierarchy is ASCL1. So if we delete ASCL1 in the mouse prostate, we can actually recover mice that have prostates that completely lack neuroendocrine cells, and yet the prostate appears to be normal, and there is a normal proximal region. So we do have a rare escaper cells in the periurethral region, which are likely due to incomplete deletion by NKX 3.1. So our current model for the origin of neuroendocrine cells is that they likely arise from a urogenal epithelial progenitor, although we haven't excluded the possibility they arise from an early luminal progenitor. But in either case, progenitor activity coincides with the developmental stages in which androgen levels are very low. And we're currently studying the molecular properties of normal neuroendocrine cells to understand, in more detail, their regulation. Moving on to cancer, we've used the NP53 mouse model to generate organoid lines that displayed neuroendocrine phenotypes. So this is work from a talented postdoc in my lab, Jia Li. She has used NP53 tumors and established a large number of organoid lines, some of which have neuroendocrine features. As you can see here, this is the sort of primary tumor, and you'll note that it's heterogeneous. These are the organoids that were established. They're green because of the lineage marking. Here's a different line that you can see the region of small cell histology. And these organoids are heterogeneous. You can see that they have a neuroendocrine region as well as a non-neuroendocrine region that is mesenchymal in nature. So this can be more clearly revealed by marker analyses. So here's a line that's relatively homogeneous. It expresses synaptophysin and chromogranin A, so it has a neuroendocrine phenotype. Here's a different line that is more heterogeneous. That has sort of mixed expression of neuroendocrine markers as well as androgen receptors. To some extent, it actually has a double positive or amphicrine phenotype. This is a heterogeneous line that I showed you earlier. So it has intermixing of neuroendocrine cells and non-neuroendocrine cells that express androgen receptor. Here is a different heterogeneous line, again, with a similar intermingling. So these neuroendocrine lines, whether they're homogeneous or heterogeneous, are highly stable during passaging. They can be passaged for more than 20 passages, and the heterogeneous lines will maintain their heterogeneity. So we can analyze the heterogeneity using single-cell RNA sequencing. And so this is the first line I showed you. And interestingly, the clusters are sort of grouped together. So we have an ARPC cluster, an NEPC cluster, and a grouping of DNPC. And this is the other heterogeneous line. And we see a similar arrangement of clusters. So the heterogeneity of these organoids is striking because it suggests that we are able to capture much of the spectrum of CRPC within organoids, established organoid lines. So what can we do with these organoid lines? Well, we can do a number of things. One thing is we can examine, sort of the epigenetic marks that are displayed in these organoid lines. So for example, we've been pursuing cut and tag analyses here, looking at H3K27 trimethyl. So this is the mark deposited by PRC2. So I'll just show you just this little tidbit here. What's interesting here is that actually the non-neuroendocrine lines appear to have a somewhat higher level of H3K27 trimethyl than the neuroendocrine lines. So that's something interesting that we're currently following up on. We've also been collaborating with Andrea Califano's laboratory, which has developed a set of computational systems approaches to identify master regulators that drive biological processes. And so one of the analytical approaches that they've developed is known as MetaViper, where we can take single-cell RNA sequencing data and analyze this to infer protein activity at the single-cell level. So Alessandro Vasievo in Andrea's lab, postdoc in Andrea's lab, has done this, and again, using the same organoid line as I showed you earlier, here by protein inference, we can see again clustering of ARPC, NEPC, and DNPC. The ARPC cluster is elevated using an androgen receptor, is enriched for an androgen receptor signature. The NEPC cluster is enriched for a neuroendocrine signature. And we can predict master regulators using this sort of approach. Notably, one of the neuroendocrine master regulators that's predicted is ASCL1. So this is a way that this is a method that we're employing to identify candidate intrinsic drivers of neuroendocrine differentiation that we're currently seeking to validate in functional assays. So one of the sort of interesting questions we can ask is, does transdifferentiation occur at some level in organoid cultures? And we have some evidence that it might. One way that we've been looking at this is using single-cell ATAC-seq to examine chromatin accessibility. And we can see that, this is the same line again, by single-cell ATAC-seq, there are 7 clusters, and these clusters have open chromatin at chromogranin A, neuroendocrine marker, these in AR. And what you can see is that there's one cluster here that has open chromatin for both chromogranin and AR. You can see this more readily, looking at the genomic locus. So this cluster 7 has accessible chromatin at both chromogranin A and AR. And so this cluster, we believe, corresponds to a potential transitional population in the process of neuroendocrine differentiation. We've also been trying to assay this directly. So this is very preliminary data, but we can isolate non-neuroendocrine cells by flow cytometry, mark them with expression of RFP, and then culture the non-neuroendocrine cells and the neuroendocrine cells separately for several passages, and they maintain their non-neuroendocrine or neuroendocrine phenotypes. If we co-culture them together, however, we now see they're rare cells that are RFP positive, but now express neuroendocrine markers such as synaptophysin or chromogranin A. And interestingly, they maintain the expression of vimentin. So these appear to be the transitional cells whose phenotype seemingly corresponds to what the ATAC-seq might predict. Okay, so these are some of the approaches that we've been employing to study lineage plasticity in prostate cancer. In the remaining 10 minutes or so, I'd like to switch over to bladder cancer and explain how we've been using organoids to study plasticity in bladder cancer. So bladder cancer is, of course, a major health problem. It's quite understudied. The normal bladder contains, again, sort of 3 epithelial cell types, as it were: basal cells, intermediate cells, and umbrella cells. And bladder cancer can be roughly divided into non-muscle invasive disease and muscle invasive disease. Historically, these have been considered to be almost distinct entities, and it's unclear what the relationship actually is. So there are 2 forms of non-muscle invasive bladder cancer: papillary and carcinoma in situ. And carcinoma in situ has been considered to be sort of the precursor to muscle invasive disease. However, there's also some evidence that papillary disease can progress to muscle invasive disease. So we've been interested in studying progression of bladder cancer, and to pursue this, we've actually established patient-derived bladder tumor organoids. And these have been established through collaboration with urologists who perform transurethral resections. So what they do is they sort of go in and extract sort of like the tops of these tumors here. This is what they actually view through the cystoscope. This might look a little uncomfortable for men in the audience, but this is how it's done. And we take these samples and we can establish organoid lines in culture, which we can serially passage. These organoids can also be grafted orthotopically. This orthotopic grafting, it uses ultrasound-guided implantation into the bladder wall, which is a very efficient process. So we can readily interconvert organoids into xenografts. We can also take the xenografts and convert them back to organoids. So all of these, together with the parental tumor, can be analyzed by sequencing or histopathology, etc. So this just shows you some of the organoid lines that we established. And what is, I think, evident is that the organoids and the xenografts retain the characteristic histology of the parental tumor. We also can establish organoid lines that have less common histological variants, such as squamous cell carcinoma. In recent unpublished work, we've now increased our biobank to approximately 50 organoid lines. We've been able to establish organoid lines from cystectomy samples as well as from transurethral resections, and have also established several lines that contain variant histologies. So in collaboration with David Sollitt's group at Memorial Sloan Kettering, and the previous pathology was in collaboration with Hikmat Alamadi at Memorial, we've analyzed these organoid lines molecularly using the targeted sequencing platform at Memorial, MSK Impact. We sequence the organoids the parental tumor in normal bloods. And we can show generally that the organoids display mutational profiles that are concordant with that of the parental tumor. We can examine sort of the mutational profiles of these organoid lines, which really recapitulates sort of the distribution of mutations in human bladder cancer. So we can see that among the common mutations, we see mutations in a lot of epigenetic regulators, which are frequently mutated in bladder cancer, such as KDM6A, KMT2C, and 2D, as well as ARID1A. Interestingly, we also see, we're also able to capture mutations that are relatively rare but interesting in bladder cancer, such as mutations in ERB2. And of note, we have very few mutations in RB. So many bladder cancer cell lines were established from metastatic bladder cancer and contain RB mutations, whereas our organoids are generally established from non-muscle invasive bladder cancer or earlier stages of muscle invasive disease and lack RB mutations. So what can we do with these organoid lines? One thing we can do is we can examine their drug response. And of particular interest, we're able to establish organoid lines from patients in a longitudinal fashion. So patients will often undergo transurethral resection, be treated, and then sometime thereafter, their tumors will unfortunately recur, and then we have an opportunity to establish another organoid line. So here's an example of a patient and a pair of organoid lines where patient was not otherwise treated. So the tumor was removed, but the patient was not otherwise treated. And we can see in terms of response to a range of different drugs that interestingly, the organoid lines display nearly overlapping drug response profiles. However, in this case, a different patient was treated with mitomycin C and BCG. The tumor relapsed after over a year. And now the recurrent organoid line is much more resistant to a range of different drugs, however, displays similar responses to other drugs. So it's of interest to us to understand how drug response has altered the properties of these organoids. So one thing we've started to do is to perform single cell analyses here of this recurrent pair. And interestingly, the recurrent organoid line is actually much more heterogeneous than the sort of the organoid line from the parental tumor. And what is interesting is if you reaggregate these together, now you can identify a cluster that is actually in common between both the first and the second organoid lines. So this cluster, we believe, corresponds to a transitional population, and we can identify markers that are specific for this transitional population. So our hope here is that we can utilize this pair of organoid lines and other examples of recurrent disease that we have to sort of replay in organoid culture the events that take place during the emergence of treatment resistance. So finally, I'd like to address the issue of tumor progression in organoids. So a non-muscle invasive disease, can be classified into two luminal categories as well as a basal category. Muscle invasive disease is more complex. And again, as I mentioned earlier, the relationship between these entities has been somewhat unclear. However, there is a question of whether, if we have progression, whether there might be a switch in subtypes, specifically from sort of class 2 luminal tumors to a basal squamous muscle invasive tumor. So we believe that we can recapitulate this in organoid culture. So many of our organoid lines are phenotypically stable in culture. If you look at different markers, they're stable in organoids as organoid xenografts and xenograft-derived organoids. However, sort of a little over a majority of the organoids from non-muscle invasive tumors display this sort of phenotypic plasticity. This is they start with a luminal phenotype here and they become basal during organoid culture. But notably, this phenotype can be largely reversed by xenografting. So we believe there's an effect of the tumor microenvironment that can repress this basal to luminal differentiation, in fact, can reverse it. But if we remove the tumors again from the xenografting culture of those organoids, they will again undergo this basal to luminal differentiation. So John Kristen, a terrific postdoc in my lab, has pursued a small molecule screen to examine organoid lines that display plasticity to see if it's possible to revert it. And he identified one big hit here, GSK-LSD1, which is a KDM1A inhibitor, and it is able to partially revert this plasticity. So I'm not showing this here, but if he knocks out KDM1A, he can also see this effect. So he is currently investigating the mechanisms by which KDM1A regulates this transition. In parallel, he's also been pursuing ATAC-seq analyses to examine sort of the epigenetic states and eventually the epigenetic marks that are associated with this plasticity. So if you examine, for example, these three organoid lines, this is a luminal line, this is a basal line, and this is what we call a plastic line. The plastic line looks like a basal line, although it started with a luminal phenotype. So we've performed ATAC-seq analyses, and we can cluster these. So you see that the luminal lines cluster together, the basal lines cluster together, and these are the plastic lines, which in the first principal component cluster together with basal, but they're separated along the second principal component. So now if we look at sort of genomic tracks, you can see that at basal markers such as keratin 14, that the basal lines, of course, have open chromatin, and the plastic lines also have open chromatin, which of course is to be expected because they have a basal phenotype. But what's interesting is that luminal markers, such as GATA3, we see that the plastic organoid lines seem to have partially open chromatin, so they retain an epigenetic memory of their luminal origin. So we are currently pursuing studies to determine whether we can actually detect this epigenetic memory in human prostate tumors, which might indicate specifically the basal squamous category, to determine whether they may have in fact originated from a more luminal tumor. Finally, we're also pursuing motif discovery approaches to identify candidate transcription factors that might be driving this transition, and we're coupling this with other computational approaches. We think that this really is modeling something that's happening during sort of transition to muscle invasive disease, because as has been noted in breast cancer for Andy Ewald, what we've found is that at the invasive front of these tumors, now we see the expression of a basal marker, cytokeratin 14, at the invasive front, but this is not expressed within the tumor body. So in closing then, I'd like to underscore that we think that these organoids represent a model system for studying tumor plasticity. So we can identify transitional populations in patients, in organoids from patients with recurrent disease. And we think that the sort of plasticity that we're studying in culture can reflect processes of disease progression in vivo. And we're using computational systems approaches to identify the drivers of plasticity. And more generally, we believe that organoid models are incredibly useful because they will allow mechanistic studies of complex questions in cancer biology that may be inaccessible using other approaches. Of course, the work that I've described involved a large team of terrific scientists who really did all the work. So notably, Laura Crowley is a graduate student in my lab who led the single cell analyses of the prostate epithelium together with Francesco Cambuli and former postdoc Mao Shibata, now at George Washington University. The work on normal neuroendocrine differentiation was performed by graduate student Ava Lung, and the prostate neuroendocrine organoids by Jia Li, a terrific postdoc in the lab. Then former lab members, Suk Lee started the bladder organoid project, and I mentioned John Kristen is a current postdoc who's played a major role in continuing this project. We've had terrific collaborators, Raul Rabada and Andrea Califano for computational systems biology, Jim McKiernan and a whole team of talented urologists who've provided samples. We've collaborated with Corey Abadi-Shen in analyses of mouse models, with Chao Lu in epigenetic analyses, and Nini Hibshush for pathology, as well as Max Loda at Cornell for pathological analyses, and a great group of collaborators at Memorial Sloan Kettering, David Solit, Hikmat Al-Ahmadi, and Barry Taylor. So thank you very much, and I'll be happy to take any questions.
Thank you very much, Michael. That was a wonderful talk and really amazing to see all of these models and everything that can be done with all of these different models. I'd like to remind the audience that they can type questions in the chat, and I will read them out and ask them to Michael. I will get started with a question, Michael. One of the things that you see in the organoids, and I was thinking in particular about the prostate cancer ones, is there's the heterogeneity. And I was just wondering whether you see shifts in the heterogeneity or of the organoids themselves when you use different types of media. So can you see shifts based on how you grow them? And then I was also wondering whether you have applied certain types of therapies to those prostate cancer organoids, and whether you see changes in that heterogeneity and shifts when you use different types of treatments.
Okay, so Katie, that's a great question. Well, questions. So the first question regarding media. So one of the things that I forgot to mention is that we use our own sort of homegrown media for all of these organoid experiments. This is a complex medium containing hepatocyte media and serum. You know, we developed this years ago to grow mouse prostate organoids. So it's quite different from the ENR-based media that many groups use to pursue organoid assays. Although you might imagine that some of the growth factors involved are in common, there are undoubtedly differences between the media compositions in terms of what's actually going on. We know that anecdotally for bladder tumor organoids that it is probably easier to establish patient-derived organoids using our media than ENR-based media. And we also know that you can transition organoid lines from one media to the other, but it may not be that straightforward. So we have some experience with ENR-based media, but all of the analyses that I've showed you today were done in our sort of homegrown media. So do we observe shifts in composition in different media? We have not really examined that, in part because it can be difficult to transition organoid lines from one media to another, media composition to another. In terms of drug treatment, we have only started to do this with respect to the prostate organoids. The bladder organoids are something that we've been examining in more detail. We've been particularly interested in mechanisms of cisplatin resistance, which of course is of considerable translational interest. So those are studies that that are, currently being pursued to examine how cisplatin treatment alters, sort of the phenotype and perhaps the heterogeneity of the organoids. But I don't have any results yet to show you.
Great, thank you. There's a question from Jeff Sklar. Have you been able to analyze human luminal bladder tumors for plasticity markers and correlate those results with subsequent development of muscle invasive tumors?
So that's a great question. Obviously, we want to extend what we've been doing in organoid culture to human specimens. So I have to confess that's just work that we've only recently gotten started. So we don't, part of the problem is actually having a cohort of patients that's suitable for this. So, we are now in the process of trying to assemble patient cohorts where we can actually have samples that are sort of longitudinal for patients who've progressed, say, from high-grade non-muscle invasive disease to muscle invasive disease. Assembling these cohorts is very non-trivial. Fortunately, we are part of a large collaboration led by Michael Shen, together with collaborators at Memorial Sloan Kettering and at Johns Hopkins. So collaborators at Memorial include David Solid and colleagues, as well as people like Jonathan Rosenberg, Dean Bajoran, Bernie Bachner, and at Johns Hopkins, led by David McConkey, Noah Hahn, and others, to try to gather together the cohorts that are essential to address this type of question, because they don't currently exist, and these types of samples are rare.
Thank you. We have another question from Mike Hurwitz, who says, great talk. Within the different luminal subtypes, do you think some are more likely to develop into cancer? Any correlate in human prostates?
Okay, so this is a question about prostate, and of course, we're very interested in cell of origin. We've always been sort of dissatisfied with our previous analyses of cell of origin because, you have luminal cells and basal cells. There's only so much you can say, but we think that there's still something to explore here because, there's this well-known phenomenon in which 85 to 90% of prostate cancer patients with sort of intermediate risk disease, will actually have indolent prostate cancer, whereas the remaining 10 to 15% of patients actually have aggressive disease. And it's difficult to distinguish between the indolent and aggressive tumors. And despite a lot of molecular analyses, they haven't tremendously improved over just simple histological Gleason grading. So we think that it remains possible that cell of origin could explain, at least partially, the difference between indolent and aggressive disease. And so that's something we're very interested in pursuing. We know already that, from the literature, that both proximal and distal luminal cells can be cells of origin in mouse models. But that does not necessarily answer the question because, they may be different in terms of their phenotype or response to treatment or ultimate outcomes. So, we're in the process of pursuing these types of studies. It's not really clear what's going on in the human prostate. And again, I think we're just scratching the surface in terms of understanding the relationship between the luminal populations in the mouse and the populations in the human. There's a lot more work that needs to be done there.
Well, thank you very much, Michael, for this visit, for this fascinating talk. I know it made me think of a lot of things. There's some parallels in the world of lung cancer, so it was really great to think about this. So thank you very much for visiting us today. And thank you everybody also who joined, and have a wonderful afternoon.
Well, thank you, Katie. Thank you, everyone.